Fermentation is incomplete oxidation reduction of glucose. This process occurs in an oxygen-deprived environment, where a cell converts glucose into either lactic acid or ethanol to produce energy. This process is also done by microbes such as yeasts and bacteria. There are two basic types of fermentation. 1. Alcoholic fermentation. 2. Lactic acid fermentation. Alcoholic fermentation by using yeast. This type of fermentation occurs in many types of yeast, for example, Saccharomyces cerevisiae. Alcoholic fermentation has great industrial importance, especially in the production of wine, beer, bread, and distilled spirits. In this process, one glucose molecule is broken down into two pyruvates. Each pyruvate is then broken into acetaldehyde and carbon dioxide. The carbon dioxide is removed in the air while acetaldehyde is reduced into ethanol. The rise of bread is due to carbon dioxide production during fermentation. Lactic acid fermentation by using bacteria. Lactic acid fermentation is caused by bacteria, where pyruvic acid is reduced into lactic acid. For example, yogurt is produced from milk by Lactobacillus bulgaricus and Streptococcus. The bacteria ferment the lactose of milk into lactic acid, thus converting milk to curd and forming yogurt. Fermentation products and their importance in daily life. Fermentation products are more likely our desired food, which make it nutritious, easily digestible, and delicious. The preserving ability of such products also increases. Thus, there is no or less need of a refrigerator. Some examples of fermented food are as under: dairy products, yogurt, and cheese. As you have already studied, yogurt is made from milk by different bacteria. Similarly, cheese is formed from milk protein by bacteria. Alcohol formation. It occurs either by bacteria or yeast. There are three main types of ethanol-containing drinks, that is wine, beer, and spirits. Wine is synthesized by fermentation of fruit juices. Mostly grape juice is used. Beer is widely used as a human drink, synthesized by fermentation of starch-containing grains like barley, rice, maize, and corn. Spirits or distilled beverages are produced by using fermented grains, fruits, and many vegetables, for example, whiskey, brandy, gin. Pickles and vegetable products. To preserve pickles and vegetable products, fermentation along with salts and acid are used. Bread formation. In bread formation, wheat dough is fermented by yeast along with bacteria. Some important industrial products produced by fermentation include glycerol, which is used for the production of plastics, soaps, cosmetics, sweeteners, and so on. Ethanol, which is used for vinegar and beverage formation and also as a solvent. Formic acid, which is used for leather treatment, rubber manufacturing, and textile dyeing. Acrylic acid, which is used for plastic making. Fermentor. A fermentor is a container in which fermentation takes place. It provides an optimum environment in which microorganisms can grow and produce commercial products on a large scale. There are many types of fermenters according to their sizes and designs. Procedure to use a fermentor. A typical fermenter contains a vessel made of stainless steel to prevent microbio-corrode and can also be cleaned easily. Microorganisms with their nutrients are put into the fermenter and air is bubbled through so that microbes can respire aerobically. As carbon dioxide builds up, the gas outlet releases it to avoid buildup of pressure. As we know, proteins are destroyed at high temperatures. Thus, to maintain optimum temperature, the fermenter is surrounded by a jacket of water. To check the internal condition of the fermenter, temperature, oxygen, and pH meters are linked with a computer. Paddle stirrers of the vessel ensure that the microbes, nutrients, and oxygen are well mixed and distribute the heat evenly. The product, which is without microbes, continuously comes out from the bottom of the vessels. Advantages of using a fermenter include: products are easily and safely separated. It is automatic, thus maintains the environment at an optimum level, meaning the external environment does not affect it. Microbes are easily put in the fermenter. Products are in bulk quantities, thus large amounts of medicines, hormones, and other products are obtained. It is inexpensive. There is less wastage of materials. It needs little space to install.